Hi, everybody. Thank you very much for having INOVIQ present to you today. I am Leearne Hinch, the CEO of the company. INOVIQ is a Melbourne-based biotechnology company that's focused on developing next-generation diagnostics and therapeutics. Could we move to the next two slides? Two slides, please. Yeah, that one. Thank you. So we have a focus on leveraging the power of exosomes to develop tests for earlier detection of cancer and also treatment of cancer. We're doing this using our patented exosome technologies, which underpin our products in market called ExoNet that are generating revenue, as well as our deep pipeline of research tools, diagnostics, and therapeutics for cancer. We've got solid data across our pipeline, and our focus is on partnering our products for commercialization with distributors, clinical laboratories, and pharmaceutical companies. The company is somewhat undervalued at the moment at $45 million, given our broad pipeline. Our key investors are Merchant Opportunities Fund, which is a biotech fund based in Australia, and also our chairman, David Williams. If we can move to the next slide. So our board and management team have extensive experience across exosome science and development and commercialization of exosome-based diagnostics and therapeutics. On the next slide, you can see that our focus is on cancer. We're using our core technologies, which we call our exosome platform, has two different types of technology. One is to isolate exosomes for use in diagnostics, and the other is to isolate exosomes at large scale, which means at low cost for use in exosome therapeutics. We have another technology called sub-B2M that detects a sugar that is only found on cancer cells. So effectively, that is a specific biomarker that we use to develop diagnostic tests or improve on existing diagnostic tests. Our exosome platform actually is the basis of our research tools our research tools segment, which has products in market generating revenue with a partner. That's about a $660 million market opportunity to provide these research tools to diagnostic and pharmaceutical companies. We have our own diagnostics in development for screening, liquid biopsies, and also development of companion diagnostics in partnership with pharmaceutical companies. And then we have an early stage therapeutics pipeline where we're effectively developing products to target and kill solid tumours. Again, very large market, and you can see it really moves to a market that's over around the $60 billion market cap. So if we can move to the next slide, please. Our product pipeline is very broad. We have our exosome research tools in market and generating revenue. We have 3 different diagnostic tests, which are based on both the sub-B2M and our exosome technologies, which I'll touch on, and our exosome therapeutics pipeline. On the next slide, you can see what is an exosome. Exosomes are essentially small packages that are released by every cell in our body. Now, what exosomes do is inside the exosome, they contain important biomarkers such as DNA, RNA, and proteins. These act as either cellular messages, which are used in therapeutics, or they are biomarkers of disease, which are used in diagnostics. INOVIQ is exploiting both their use for diagnostics and therapeutics in our pipeline. On the next slide, you'll see our core ExoNet exosome isolation technology. It is a best-in-class capture tool for exosomes, which is now in market, generating revenues with a partner called Promega. The way the technology works is it is a magnetic bead-based technology where effectively we isolate exosomes based on the surface proteins which are found on the exosome. So we use a variety of different antibodies to pull out exosomes from different tissues in the body. This can be done at large scale, meaning that we can essentially do over 1,000 samples per day using this technology. Existing exosome capture technologies in the market, the majority of them are only used for research purposes. We have developed this tool to enable a concept all the way through to the clinic, meaning a diagnostic test in a commercial lab that will actually perform at scale. Our technology offers speed, efficiency, and scalability. We've got data published that validates our technology in cancer, neurodegenerative disease such as Alzheimer's and other diseases. And we can also develop sample exosomes from the brain and also exosomes that come from tumours. That enables us then to find specific biomarkers for these diseases. On the next slide, we show that we have a partnership with a company called Promega Corporation. They are a multinational company providing services to the life science industry. They have over 2,000 products on the market. They partnered with INOVIQ in order to utilize our exosome capture technology, which then enables a range of products that they have for isolating what's inside the exosome, meaning the RNA as an example. Our agreement with them is a non-exclusive agreement to distribute our product worldwide, and they essentially will underpin the growth of our ExoNet sales of this product going forward. We signed this agreement in April 24, and the company is very excited about this partnership. Additionally, we as a company provide exosome services to pharmaceutical companies, and we have our first partner that we're developing a potential companion diagnostic with. A companion diagnostic is essentially a test that's being used to select patients that will respond to an immunotherapeutic, a very important area of the company's pipeline. Moving on to the next slide. What we're seeking to do with our exosome technology is to develop our own diagnostics and therapeutics pipeline. We have both in-house products, with our lead product being an ovarian cancer test for screening or early detection of ovarian cancer in high-risk women. As many of you know, there are no tests available to detect ovarian cancer early. So this is a huge unmet need and a potentially very large market. Which spans from, if we're looking at the high-risk women, which is women that have mutations to BRCA1 and BRCA2 genes, that's effectively around a $300 million market for high-risk women alone. If all women were screened over the age of 50, this would be a $32 billion market. So you can see this is a very large market with an unmet need, and this is what the company is focused on. In terms of the next slide, this takes us on to our exosome therapeutics program. What we're doing here is developing weaponized exosomes. These essentially are an off-the-shelf treatment to target and kill solid tumors. What they do is we take a CAR T cell or a CAR NK, natural killer cell, which you're probably aware is some of the key therapies out there for treating hematological or blood cancers. We take these cells, which have a CAR, so an antigen on their surface that can be used to target, for example, breast cancer. The exosomes that are released from these breast cancer cells inherit the same property as their parent cells. So they contain both this CAR antigen for targeting breast cancer, and also they have the same cytotoxic properties as their parent natural killer cell. So they then these exosomes target breast cancer and kill the breast cancer cells. And we have shown that we can recover the exosomes produced from CAR T cells, with 80% of them can be recovered with 95% purity. Additionally, we kill 75% of breast cancer cells in vitro within 3 days. So that's an extremely positive result, and the company is now moving into preclinical in vitro and in vivo studies, where we expect to report data in calendar year, later this year, I should say, and next year. Importantly, effectively, exosome therapeutics have some significant advantages over autologous CAR T cells in that they have both safety advantages and potentially efficacy advantages for treatment of solid tumours. If I can jump to the next slide, I'll just briefly touch on our sub-B2M technology. That's our other technology used for detect. Our lead project there is our breast cancer project. It's being developed to improve on an existing test used for monitoring breast cancer. The advantage of this test is it improves both sensitivity, on the next slide, which is the ability to detect disease, as well as specificity of disease. And what we've shown is when we compared this test to the existing test on market by Roche Diagnostics, we showed an increased sensitivity from their 37% to 81% of cancers being detected, and an improved specificity. So we've completed both a clinical validation study across all stages of breast cancer, so early stage through to late stage disease, as well as a monitoring study where we showed equivalence. Ultimately, we expect to launch this product in the first half of calendar year 25 with a clinical laboratory partner. If I can move to the next slide. In summary, the company is now well positioned for growth. We have a proprietary exosome platform across research, diagnostic, and therapeutic applications. We have commercial partners secured for ExoNet that's now generating revenues, and we expect to be able to demonstrate recurring revenues for ExoNet moving forward. We are earning service fees for providing services to pharmaceutical companies to develop companion diagnostics. We've got considerable strong data for our tests in development, so particularly our breast cancer tests, which we expect to commercialise early next year, and a strong pipeline of diagnostics and therapeutics. And on the next slide, you'll see that we have a lot of milestones coming up that the company expects to deliver on over the next 6 to 12 months, including revenues for our ExoNet technology in market, an agreement with a pharmaceutical company around developing a companion diagnostic for exosomes, and in addition, advancing our own ovarian cancer diagnostic screening test. And on the sub-B2M side, as I've already mentioned, we expect to launch that test with a commercial partner in the first half of next year. So the company is in a very strong position in terms of its pipeline, and we look forward to hopefully having some investment from some of you in the near future. Thank you.